Hello, hello, hello. How are we today?
Tired.
Tired? I'm tired too. I am very tired too, but biochem, we love it. All right, as always, please sign in first. Just get that out of the way really quickly. Today shouldn't be too bad of a day, so that's exciting. Do y'all have cells and tissues tomorrow? Anything like cells and tissues related?
No, not
Well, we have a mock practical next Tuesday, but we have a practice mock practical.
A practice mock practical.
Yeah, the practice.
Okay.
We have open left.
Okay. Cool. So we'll just have this up here for a little bit.
It's a practice mock.
Practice mock practical.
Where is Dr. Sarkar at currently in lecture? Has he? Good. He actually went back and did that? Good. I talked to him, and so he said he would do that, so that's wonderful. So he's still going lecturing chapter 5? Or okay. Okay. Do y'all want me to do the end of chapter 4 again? That's already been done. If we can just plow ahead, that would be perfect. Okay, does anybody need this up here still? Cool, I'm going to switch over. All right, chapter 6, not too bad. There's really just a few topics that we need to go over, but they're kind of repeated throughout. So really not too bad. Chapter 7 gets a little more chemistry. We kind of return to that beautiful, glorious land of chemistry that we love so much, or I do at least.
Are we skipping chapter 5?
We did chapter 5 last week.
Oh, okay.
So chapter 6. So yeah, I'm actually ahead of him, which is rare. So let's just kind of keep that up, yes or no? Yeah. Let's keep that up. Let's be prepared. Cool. All right. Chapter 6, talking about the regulation of fuel metabolism by insulin, glucagon, and other hormones. All right. So where do we usually hear insulin coming into play in the real world? Diabetes, right? We have the two different types of diabetes. All right. We have 1 and 2. And type 1 is when the body doesn't produce enough insulin itself. There's not enough endogenous insulin. So typically there has to be some supplementation of that. So that's when we get the injections. Whereas in type 2 diabetes, that's where we have our diet is high in sugar, but actually more importantly high in fat, and our insulin cells are being, they're decreased sensitivity to the uptake of sugar. So pretty much we just fatigue the amount of insulin that we can make, and therefore lowering the ability to draw sugar into the cell, leading to high blood sugar. And that's what that is. So there's the two different types. We're not going to go into that in here, but kind of understanding that physiology, important. So starting off, we're going to be talking about glucose, glycogen for the rest of this chapter and kind of forever. So understanding the differences between glucose and glycogen is going to be highly important. So glycogen, storage form of glucose. All right, so there's multiple It's pretty much just a chain or multiple chains of glucose, and it's the storage form. It's branched, makes it easier to store, what have you. Glucose is just the monomer, the basic building block. It's our simple sugar. So just understanding that. So whenever we're talking about glucose, we're just talking about that single glucose molecule. And if we want to see a picture of it, glucose. That's the basic structure of glucose, and a bunch of those would make up glycogen. All right, so we have three major nutrients that our body uses, right? We all know that we use carbs, fats, proteins. Yes? If not, we learned something today, so hooray for you. So we have carbs, fats, and proteins. And all those help provide energy for us to live and to maintain homeostasis, right? So that's why we eat, so that we can keep living. If we stop eating, we will stop living. And so the fuels that we actually use are the glucose, free fatty acids, glycerol, amino acids. We'll get to those.
Andrew?
Yes, sir.
I thought you said we were doing bioenergetics, and I might have missed something that you were talking about. But I thought next lecture was supposed to be bioenergetics, chapter 7.
Yeah, well, we haven't covered chapter 6 yet.
Okay, so he's going to skip there, or he's going to go over chapter 6?
He should do chapter 6 and then chapter 7. Yeah,
So he's doing chapter 6 and chapter 7.
he should be,
Okay.
as far as I'm aware. I don't think he's ever skipped chapter 6,
Okay. Said,
but he might.
I thought I heard at the end of his lecture that he said he was doing bioenergetics next, but you couldn't totally understand.
Well, we're going to do chapter 6 and 7 today, so we're going to
Oh, we're
do
doing
both.
Okay.
Cool. All right, so that's pretty much basics. We've seen, going to the next page, we've seen our classes of hormones, right? That's on page 75 previously, so we've already seen this, right? We learned the difference between plasma membrane receptors and nuclear membrane receptors. Which ones work faster? Plasma membrane receptors, right? Because the hormone doesn't have to travel all the way through that plasma membrane. It doesn't have to travel through the nuclear membrane to actually get to the DNA. The plasma membrane receptor, it just binds on the outside of that cell, causing that G protein coupled receptor to amplify that signal using what as the second messenger? Cyclic AMP, right? Cyclic AMP is a second messenger. Should we go over that real quick before? Should we go over that one more time? All right, let's go over that one more time. Gotta know it, gotta know it. How was that assignment? Will I help y'all with it? No. I cannot do that, unfortunately. All right, so mechanisms of action for our G protein coupled receptor. All right, so first thing, ligand binds to the receptor, right? So over here, first thing that's got to happen is we have ligand binding to the receptor. We have our signal, receptor, transducer, effector response. Got to know that order. Ligand binds to receptor. Signals transmitted to our G proteins, and our G protein has multiple subunits, right? We have our alpha, beta, and gamma. The inactive form of the G protein has the GDP bound to it, right? So it's inactive, that GDP is bound. And which subunit is it bound to? Alpha, right? It's bound to that alpha subunit. So what happens is when that ligand binds, GTP comes in and knocks out or replaces that GDP on that alpha subunit, right? So we have that, I guess, GTP comes in and knocks out, there's a GDP on there. And that causes, when that GTP binds, that causes the alpha subunit to separate from the beta and gamma subunits, right? So we started off, they were all together, and now GTP comes in, binds, splits it apart. We'll get back to the GTPase here in a second. All right, so that alpha subunit, that can either be a stimulatory subunit, so it can activate something else, or it can be an inhibitory, so that can go on and turn something off. That's all it's saying. For the rest of this, until I think the very last thing is all the stimulatory, just to get that straightened away. So this activated alpha subunit now goes and activates adenylate cyclase. That's this squiggly little thingy over here. That goes and activates that. Adenylate cyclase produces cyclic AMP, right? So we need to activate this adenylate cyclase so that we can produce our second messenger, which will then relay the rest of that signal to the rest of the cell and amplify it. All right, so it's like a marathon runner. Like this is the first leg, right? And then our cyclic AMP is like the second person, or I guess it'd be like a relay. It'd be like a team relay, right? So first he's got the baton, he's got the signal, and then cyclic AMP finishes out, amplifies it. All right, so we've made cyclic AMP. Adenylate cyclase produces cyclic AMP. Where does that go? Cyclic AMP converts an inactive protein kinase to active protein kinase. The function of protein kinase is to cause protein phosphorylation. So this protein kinase has 2 regulatory subunits on it and 2 catalytic subunits, or active ones. So what cyclic AMP does is it binds to the regulatory subunits, removes them, takes them out, right? So that the catalytic ones can go on and do their job. Where are we at? The substrate protein is dephosphorylated by another enzyme that removes the phosphate group and is known as protein phosphatase. This is where I always get lost, every time. So I'm sure y'all do too. So cyclic AMP binds, removes. Catalytic free can cause protein phosphorylation. Removes the phosphate group and is known as protein. So this protein phosphatase is all it's saying is that after the catalytic enzymes add a phosphate on there, they cause protein phosphorylation. So phosphor equal phosphate. Ylation is just saying I'm adding a phosphate onto it. All this is saying is that protein phosphatase can then go and, take that phosphate back off. So this is kind of, let's just mark kind of, this is saying it's reversing. Reversing action of protein kinase. And so it's saying that sometimes so there's a series of reactions where it puts the phosphate on, puts the phosphate off. And then when we put that phosphate on, biological response happens. Something in the body changes, it's different. Action has been completed. So if we want to turn it off, if we're done with what we need to do in our body, we can either break down or destroy or inactivate our cyclic AMP, which is our second messenger. So cyclic AMP. And this cyclic nucleotide phosphodiesterase is what converts cyclic AMP to the non-cyclic AMP form. Structure determines what? Function, right? So cyclic is is circular. So if we turn cyclic AMP into linear AMP, is that going to be active or inactive? Inactive, right? Because it has to have that circular shape to do its job. So that's all it's saying is that this enzyme is turning AMP into its non-cyclic form, thereby preventing it from doing its job, carrying the message. And here's our GI. The GI stands for inhibitory. So instead of this alpha subunit going over and activating adenylate cyclase, it's going over and it's inhibiting it. And so thereby, if adenylate cyclase is inhibited, it can't produce cyclic AMP, right? That's what produces cyclic AMP. So if we turn that power button off, can't produce it, shuts off the response of whatever that second messenger system's doing. So that's a lot. It will only be on the final a little bit. That's just a very broad general term. A lot of that assignment is probably, oh shoot, is the batteries working or something? Batteries are back working, good. All right, so we said our second messenger is cyclic AMP. Calcium is our other big one, mainly cyclic AMP. But knowing ligand binds, GTP comes in, knocks out GDP, attaches to the alpha subunit. Alpha subunit goes, attaches to adenylate cyclase. Adenylate cyclase is turned on, right? We turn that power button on. That produces cyclic AMP. Cyclic AMP then goes and binds to the regulatory subunit protein kinase, removes it, leaving just the ones that work. Those that work go in phosphorylated protein, activating it, causing them to turn on to go do whatever they need to do. And then we have our functional target protein. And then we can either destroy cyclic AMP, we can destroy the second messenger to where that shuts it off, or we can take off the GTP here, or we can remove the ligand up here, and any one of those can reverse what we just did. Right? So our body is constantly doing things, taking it back, doing things, taking it back. That's just like bare bones what that is. There was a link in last week's podcast of Khan Academy I thought was pretty good. So if y'all want to watch that again too. Cool. All right. Glucose is our most important, I would say it's our most important energy source that we have for our body. Our brain actually works pretty much exclusively on glucose. So that's why we like things that are sugary and chocolatey and sweet, right? Because our brain says, oh, I use that as energy, so I'm going to try to make that taste good so that I keep eating it. And also, what is it? In order to break down fats, we actually have to have carbohydrates in our body to start that from happening. So we can't break down fats without first using carbohydrates. Kind of cool. You'll learn that in nutrition. All right, so 3 things: carbs, fats, proteins. There is one molecule that links all 3 of those together, and that is acetyl coenzyme A, or acetyl-CoA. That's probably a test question. If not, it's for sure going to be a quiz question on y'all's next quiz. So get that one right. So we talk about how, like bodybuilders, they try to eat a lot of protein, right, so they can build muscle. Or marathon runners are going to carbo load beforehand, before they run. And people who eat fats try to eat the good ones, right? Don't eat the trans ones, whatever. So, but there's acetyl-CoA is the one intermediate to where no matter if you have a high fat diet or a high carb diet or a high protein diet, whatever it is, those can all come together and make acetyl-CoA. So that's really nice to where, say that we do need more proteins, say I guess we have too high of a fat diet, our body can convert it to acetyl-CoA, and then acetyl-CoA can go and help make proteins because we're not getting enough. So it's kind of like an intermediate that can swap in between everything. Very, very important. And the liver is the major tissue for fuel metabolism, also a test question. For a lot of biochemistry, when in doubt, the liver is probably going to be the right answer. So a lot of our metabolism happens in our liver. Okay, so we talked about insulin, glucagon a little bit earlier. Insulin, let's see if that's over here. So we have our pancreatic hormones, our pancreas, right? We know where our pancreas is located at. Maybe not. We'll learn it eventually. All right, so pancreas produces our insulin and glucagon. And we also have our adrenal hormones, which are epinephrine, norepinephrine, cortisol. Cortisol, we'll get to it on the next page. Yeah, it's all like on the next page. I don't really know why he has it all just like here. All those affect our blood glucose levels. So if our blood glucose levels are too high, insulin is going to signal to the tissues that we need to bring it into the cell, right? We need to have, y'all learn in lab die, also try 5. It's amazing what happens, like when you hit try 5, everything just kind of starts clicking. So y'all can look forward to that. There's normal levels for everything in our blood and our body, what have you. If they're too high, the body is going to regulate it and say, hey, we need to drop these levels down. If they're too low, vice versa. So if our blood sugar is too high, we're going to pump out insulin to bring up that excess glucose, right? Diabetes. Either you don't have that insulin, type A or type 1, or your insulin receptors are resistant to, they're pretty much, it's like they cried wolf so many times. Like you keep saying, like, they're fatigued, that's the word. They're fatigued. So you keep pumping out this insulin, like they're worn out, they're broken down. It's like finals week. You just can't do anything. You can't move. You just want to stay in bed and watch Netflix all day, every day. Like you just can't do it. So that's what happens with type 2 diabetes. And then it brings in that glucose and converts it to the storage form as glycogen, or it can convert it into fatty acids. So glycogen, fatty acids, storage. That's why we put on fat, right? Because it's just a storage form. We're eating too much. The body can't use it all, so it just stores it as fat. Glucagon does the opposite. So glucagon is signals cells when the blood glucose is too low. So insulin, blood glucose is too high. We need to bring that glucose out of the blood into the cells to store it. Glucagon there's not enough glucose or sugar in the blood, so we need to take it from storage and put it back in there for us to use. Important word here, a couple words, glycogenolysis. It says glycogen, which means glycogen, which is nice. And then whenever you see lysis, is the breakdown. So pretty much we're breaking down glycogen and making glucose. And we also have gluconeogenesis. So those are kind of the two ones that we'll need to differentiate here. Gluco, meaning glucose. Neo, meaning new. Genesis, meaning the creation of. So what happens is we're making glucose pretty much from non-carbohydrate sources. So we're going to, in some form or fashion, convert either proteins or fats into acetyl-CoA or some other intermediate that can then lead to glucose. So it's using either proteins or fats to make glucose. Cool. Epinephrine released into the blood to prepare muscles for a quick burst of activity, right? Fight or flight. So epinephrine is released, and that kind of just preps everything preps our body to be able to have quick bursts of energy, whereas our cortisol is our response to long-term stresses, right? We don't want long-term stresses, correct? Yes? But that's what we are all as humans like constantly in a state of, right? So as chiropractors, we have huge impacts on the autonomic nervous system. So if we can, with that adjusted thrust, we can actually decrease cortisol levels, putting them into parasympathetic, which is immensely helpful because they're never in that state. If we can just train that body to do that, it's a beautiful thing, beautiful thing. Insulin, glucagon, antagonistic to each other. They are opposites, right? One takes up glucose from the blood, the other one puts glucose into the blood. You need to know the difference here between pancreatic beta cells and alpha cells. It'll come back for boards. They'll ask you this. So pancreatic beta cells from the islets of Langerhans secrete insulin. So beta cells from the pancreas secrete insulin, whereas the alpha cells secrete glucagon. Need to know that. Cool. And so this is just saying high blood glucose, insulin secreted. Insulin tells the cells to uptake that glucose, normalizing blood glucose levels. It's taken up mostly in the muscle and our adipose tissue, and they're converted to glucose 6-phosphate, and that is actually the first step of glycolysis by the hexokinase enzyme. Remember that one? Hexo meaning 6, kinase meaning phosphate. We'll get to it. Insulin is not always in its active form, right? The body does not always have everything in its active form. Actually, with a lot of our supplements that we take or that our patients are going to take, a lot of times they'll be either in its active form or inactive form. So if you ever see anything that says methyl in front of it, that means it's in its active form. So your body has to do less work to actually convert that into its active form. So it's just good to go. But if there's anything besides methyl in front of it, the body actually, you're going to actually have to add an extra step for that enzyme or for that vitamin to be used by the body. So along those same principles, insulin is not always at this mature state. It does have to go through some steps to get to its active form. And we need to know the order, pretty much just the order of this, that it goes pro-insulin, mature insulin. And I think that one's pretty straightforward. This one has pre-pro, that one just has pro, that one has mature. Not that big of a deal. So I'm going to keep going. The biggest one that we're going to talk about is insulin, and glucagon is going to have pretty much the opposite effect that insulin is going to have. So metabolic effects of insulin. Increase glucose uptake, right? We've kind of just said that 12 times already. It's going to increase glucose uptake. It's going to increase our glycogen synthesis. Glycogen is the what form of glucose? The storage form, right? So if we keep taking it in, we're going to need to store it as something. So if we're increasing our glucose uptake, it's going to have to go somewhere, so it's going to get stored as glycogen. So if we're making glycogen, we don't need to be breaking down glycogen, correct? That just makes sense. If we're adding more, we don't need to be breaking it down because we're storing it. We are increasing our glycolysis, so glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate, which y'all will learn here very, very soon, is glycolysis. So the body is going to use those sugars, that glucose, to actually, like for our brain to work, for our muscles to be able to perform their actions. So we're going to increase the breakdown of glucose, which is glycolysis. We're going to increase our fatty acid synthesis in the liver, right? Like I said, it's got to be stored somewhere, and by default also our triglyceride synthesis. And these are all the enzymes. So for glucagon, we would just say the exact all of this would be the opposite, because they are antagonistic to each other. Y'all don't need, this is just kind of like understanding. Probably this half of the chart is going to be a lot more important. I don't think he's going to be asking you about these enzymes quite yet. So we'll get to those here in the next few chapters. This kind of just broad overview before we get into glycolysis, Krebs, oxidative phosphorylation. Insulin is a tyrosine kinase type receptor. So going back to the previous chapter, we had our different types, right? We had our GPCRs, our nuclear membrane receptors, and I think our tyrosine kinase receptors, correct? Does anybody remember any of that? Where is that at? Where is that at? It's somewhere. Anyway, regardless, insulin, tyrosine kinase, I've seen that show up. Whereas chapter 5, it was chapter 5. What page? So close. Receptors with enzymatic activities, insulin receptor, tyrosine kinase. Look at that, even showed up as the example. So it's a tyrosine kinase. So we've already seen that in the 2 chapters now. Just an easy, did you read the chapter or not question. Tyrosine kinase, whereas glucagon uses that G protein coupled receptor that we went through that whole example for just 15 minutes ago. So glucagon, now we've got glucagon. That's when we have low blood sugar. And so if we have low blood sugar, we're going to increase our glucagon. So by default, we're going to decrease our insulin release, right? Because we don't want to be having sugar being taken from the blood when we need to be pumping it into it. So when glucagon levels are increased, our insulin levels are going to be decreased. So here's kind of the opposite of what's happening on this other side here. So we're going to stimulate glycogen breakdown in our liver because we need that glycogen, that storage form. It's Christmas. We need to take our decorations out of the closet, out of storage, and put them up for everyone to see. So it's breaking down glycogen into glucose. It's activating glycogen phosphorylase and inactivating glycogen synthase. All that's saying is glycogen phosphorylase is what breaks down glycogen, and glycogen synthase is what makes glycogen. So it's just saying we're going to activate the one that breaks it down, and we're going to start turning some off that makes glycogen. We're going to inhibit glucose breakdown by glycolysis. So we don't want to use glucose. We want to keep that in our blood sugar. We don't want our cells to have this be absorbed and to take it through glycolysis. So we're going to turn off glycolysis. We're going to stimulate gluconeogenesis. Gluco meaning glucose, neo being new, genesis meaning the creation of. So we need more glucose, so we got to make it. We don't have enough glycogen, so we're going to eat it from a non-carbohydrate source, whether that be fats or proteins, what have you. And we're going to break down our fats, right? So we can turn our fats into that acetyl-CoA, and that acetyl-CoA can then, in turn, be made into glucose, right? So acetyl-CoA as that mediator between all 3 of these, huge, huge, huge. And increasing ketogenesis, ketone bodies can be used in like emergency situations, but long term, not very good at all. We'll get to that. We'll get to it. But that's just opposites. Glucagon, just G protein coupled reaction receptors. So if you ever see that, you're going to think cyclic AMP, protein kinase, protein phosphorylation. And all right, so flow chart comparing the 2. So if we're eating we're increasing our blood glucose. Our pancreatic islet cells of Langerhans, which one's alpha or beta? Beta. Our beta ones are going to secrete insulin. Insulin's going to cause the cellular uptake of glucose, so our blood glucose is going to go down, right, back to normal levels or what have you. And so when blood glucose levels go back down to normal, we have a negative feedback control that stops insulin from taking in that glucose from the blood sugar, right? So if it just kept going and going and going, we'd have none left in our blood. Not good. So there's got to be some kind of feedback saying, hey, we've got enough. We're back down to homeostasis. We're at normal levels. Let's just stay there. Don't go past it. And that's the same for glucagon. So in our fed states, in our eating states, we're going to be using insulin. But in our starving or our fasting states, we're going to typically see more glucagon. And that's chapter 6. Just all glucagon, insulin. Where is it made? How does it work? Take 10 minutes. Palpate. All right. Come back. We got just a... So chapter 7 is our bioenergetics chapter. We're going to do chapter 7, right? Let's overachieve. Let's do it. Chapter 7 is our bioenergetics. So like I said earlier, this is kind of where we get back into a little bit of that chemistry. And so for those of you who haven't had chemistry in a while, there's some definitions that might seem a little bit unfamiliar to you. So I was looking today, not in class, because that would be irresponsible, for some videos that could help. And this is the best one that I found. So we're just going to watch this nice little video.
Welcome to my podcast on Gibbs free energy. Whenever I get to Gibbs free energy in the year, I can see my kids' eyes gloss over and they're confused. And even William Gibbs talks about this in this quote. He said, "The idea of entropy and the second law of thermodynamics will seem far-fetched and may repel beginners as obscure and difficult for comprehension." So what does that mean? I'm going to try to tackle something that's been a problem for years. I'm going to try to make Gibbs free energy more understandable, and so that you really know what it is and understand the power of it, especially in biology. And so I think the first place to start is the name itself. The idea of free kind of gets in the way. And so what I'd like to go back to is the old name, which it used to be called available energy. And so let's think of delta G as available energy, or energy that we can actually use to do work. The other thing that I think makes it hard is the equation itself is confusing. We've got delta G, we've got enthalpy, we've got entropy, we've got words that you're maybe not understanding. And so what I'm going to try to do is pare this down to the easiest explanation that I can, and then we can get into the more of the specifics a little bit later. And so the best place to start when you're trying to understand available energy or free energy is to look at spontaneous reactions. Spontaneous reactions are reactions that once you give them a little bit of a push, they'll actually go on their own. These reactions also will tend to release energy, and they give energy to their surroundings. Quintessential example in life is cellular respiration. But let's not get there yet. So the three reactions that I'm going to talk about here are, number one, a ball rolling down a slide. Number two, diffusion, and then number three, a cherry bomb that's exploding. And so we're going to apply Gibbs free energy to each of these. So let's start with the ball at the top of the slide, and the ball at the top of the slide is going to roll down to the bottom. And so the first thing that I want to give you is what's called enthalpy, or total energy of a system. And so if we say this is a system, what happens to the total energy of the system as the ball rolls down the slide? Well, if you know anything about potential energy, the amount of potential energy that we have up here is going to be greater than the amount of potential energy that we have down here. And so the total energy of the system, we'll call that H, or enthalpy. The total energy of the system has gone from a big H to a little h. In other words, the total energy of the system has decreased when the ball rolls down. Now, if I had to push it up again, then we would add energy to the system. But this is a spontaneous reaction, and the enthalpy, or the total energy, is decreasing. Now, in biology, we don't move around because of potential energy or mechanical potential energy. In biology, our energy, or our potential energy, is actually in these bonds. In other words, there's a huge amount of potential energy in this bond between the carbon and the hydrogen. And so this right here is glucose. And if we can release some of that energy, we can do it to do work. And so again, what is H? H stands for the total energy of the system. And it looks like in a spontaneous reaction, that's actually going to get smaller or decrease. Okay, next one, let's talk about diffusion. So in diffusion, imagine that right here we've got a bunch of molecules in this container and they're bouncing around, and I remove this wall. So if I remove that wall, what's going to happen to the molecules? The molecules are going to spread out to fill that area. We call that diffusion. Now entropy is, we use the symbol S for that. Entropy is a measure of the disorder of a system, or sometimes we call that the randomness of the system. And so let's compare this. Right here we've got a bunch of molecules on this side, and then we've got a lot of space over here. So what happens to the disorder of that system as I do diffusion? Well, it's becoming more disordered. In other words, the entropy is increasing. What's an example of that? Let's say I go into your room and your room's a mess. It looks like this. If I say clean up your room, then you could go like that. And so what happens through diffusion, or in this spontaneous reaction? Well, let's say remove this wall. What's going to happen now? We're going to get even more disorder. So we're going to even get a bigger S value. And so in this spontaneous reaction, it looks like the S value is increasing. Okay, the last one is that cherry bomb. So let's say we have a cherry bomb. We put it on the desk. Does it explode? No. And one of the reasons it doesn't is the temperature is really low. And so let's say I add a bonfire to the situation, so I increase the temperature. Does that make it a more spontaneous reaction, or is the reaction more likely to happen or less? Well, it's more likely to happen if I increase the temperature, more likely to get an explosion. Okay, so those three things, total energy or enthalpy, entropy or S, and temperature can affect spontaneous reactions. And so now let's apply that to Gibbs free energy. And so before we actually get to the equation, let's just do a little algebra here. So let's say I wrote this equation, x equals y minus ab. Okay, so let's say we had this equation right here, and I were to decrease this value, the y value. What would that do to the x value? It would decrease it, right? Let's say I were to increase the a value and increase the b value. What would that do to the x value? Well, since we're subtracting right here, that would decrease it as well, or make it even go farther down. And so let's go back and summarize those three spontaneous reactions. In the first one, the ball rolled down. So what happened to our H value? Well, our enthalpy of the system decreased. What should that do to our Gibbs free energy or available energy? It should decrease that value. What happened here when we increase the entropy of the system, or increase the delta S? Well, if we increase the delta S, that should also decrease the Gibbs free energy. So now we have two things decreasing that. And what happened here? Well, if we increase the temperature, that made it more spontaneous. So if we increase that, we also decrease the Gibbs free energy. So what's the moral of the story? Moral of the story is that if the delta G ever decreases, or if it's ever less than 0, that's a spontaneous reaction. Likewise, if the delta G is greater than 0, let's turn to the next slide, that's going to not be a non-spontaneous reaction. Okay, so in summary, if the delta G is less than 0, that's a spontaneous reaction, or we call that an exergonic reaction, or an energy-releasing reaction. If it's greater than 0, that's an endergonic reaction. And then finally, if nothing happens to the available free energy, then it's just at equilibrium. The quintessential example of a spontaneous reaction in life is going to be cellular respiration. Photosynthesis is an example of an endergonic reaction. And so let's talk about each of those. So let's say we're doing cellular respiration. So what I said earlier is that glucose has a certain amount of energy within the bonds. And so let's do cellular respiration. So I take in sugar, my body is going to combine that with oxygen, we're going to convert that to carbon dioxide and water. So let's animate and see what happens. Wow, so a lot of stuff happens. So let's go back. So first of all, let's look at the entropy of the system. What happened? Did it become more or less ordered? Seems like it became more random, for sure. What happened to the enthalpy of the system? Well, we're releasing energy. So we have energy out here, so we've released energy. That means that the total energy that was contained within those bonds has actually decreased. And so you can look at the value right down here. The delta G of cellular respiration is negative 686 kilocalories per mole. That means if you had about 1/3 of a pound of glucose or sugar, you were to break that down in your body, you would release about 686 calories, big C, of energy. So we call that an exergonic reaction, or it's releasing energy. Now we can actually plot that on a energy diagram. So glucose itself had a certain amount of free energy to start with, but in this cellular respiration, we end up with less energy. So this is a spontaneous or an exergonic reaction. Now, it doesn't just explode into flames, the sugar that's sitting on your counter or the sugar that's inside your body. And so in order to get that to work, you have to put a certain amount of energy into the system. And that amount of energy we call activation energy. And so if you look back to the first three examples I gave you, the ball just didn't roll down the slide on its own. You had to push it. And the cherry bomb just didn't explode on its own. You had to add a little bit of energy to that. We call that activation energy. But there's a net loss in energy, and so we call that an exergonic reaction. Let's think about photosynthesis. What do you need for photosynthesis? Really only three things. Carbon dioxide, water, and sunlight. Oh, four things, because you also need plants. And so in photosynthesis, what happens? Well, we store that energy in glucose. And so if we look at our delta G value, it's a positive, or we call that an endergonic reaction. If we were to actually draw the energy diagram of that, we have a lower amount of energy to start with, a greater amount of energy at the end. And so we're storing energy, and then it has more energy at the end in the form of that glucose. Now, where do we get the activation energy? In photosynthesis, we get that from the sun. And so those two reactions, releasing energy and storing energy, allow life to exist. But our day-to-day life doesn't use glucose, doesn't use, or we do use glucose, but our second-to-second kind of a functioning doesn't use glucose. We actually use something called ATP. And so ATP, or adenosine triphosphate, is the energy coinage of our cells. It's what you're using right now. We can store it as energy, and then we can kind of cash it in. So let me show you what happens. ATP is broken down into ADP, and it releases a certain amount of energy. If we look at that, the breakdown of ATP to ADP, or adenosine diphosphate, it only has two phosphates right here, plus a phosphate group, is a exergonic, or the delta G value is going to be negative. If we go back and convert that ADP back into ATP, now you can see right here that our delta G value is actually positive. Or if we break it, we release energy, and then we can store that again when we make ATP again. And so right now, when you move your finger like this, what's actually powering that muscle is ATP releasing energy. It allows that muscle to contract, and then we can do that over and over and over again. And so you use respiration to get energy, but we actually store that in ATP over time. So what is the secret of life, and how does all of this work? Well, energy comes from the sun. So we have a certain amount of energy up here, and we store that energy through photosynthesis in sugars. And so the release of energy by the sun, the delta G is going to be less than zero. It's an exergonic, or an energy releasing reaction. But photosynthesis is going to have a delta G value that is greater than zero. In other words, it requires energy, and it pairs that to the energy coming from the sun. Next, we make bread out of that, and then through cellular respiration, we are going to release energy. So the delta G value now is less than zero. We use that to store ATP. Now that ATP is used to break down into ADP, and so that's going to be a delta G value of less than zero, and that's going to release energy. But to convert that ADP back into ATP is going to be a delta G value greater than zero. And so available energy and free energy is super important. It's converting energy in the sun eventually into energy that we can use, and eventually that energy ends as heat. So the energy is converted that whole way through, but it just becomes more and more disordered as we get to the end. And so that's free energy, and I hope that's helpful.
That's very, before we start anything, we won't have to do any math. So don't worry about having to do math, make numbers. Don't worry about that. Key things from that, from what he just said, are going to be found in here. We're on page, what page are we on? We're on page 91. You don't believe we've already gone through 90 pages of the textbook, right? And we've read them all. Can you believe we've read a textbook? Oh my gosh. All right, so our Gibbs free energy is our useful energy. It's our energy that can be converted to do work within our body. We're constantly making bonds, breaking bonds, using energy, releasing energy. And so they've come up with words to describe those types of reactions. So our exergonic reactions are when energy is released. So when we lose energy, so that's when we're breaking a chemical bond, releasing that potential energy. So energy is exiting. Exergonic energy is exiting. Whereas if we're making bonds or we're putting energy into a system or into, what was the example that he used? He said like photosynthesis. Those are going to be our endergonic. Energy is entering. And pretty much all of our life, our metabolism, is just the interplay between endergonic and exergonic reactions. So that's the secret of life, is just metabolism and things that release and make energy. So some people go climb to the top of a mountain and ask people for that kind of stuff, but you just get it right here. As Dr. Cusano would say in Try 5, that was a joke. You can laugh. All right, so our exergonic energy leaving, endergonic entering system. Cool. So like we said, you're going to have to know the definition of Gibbs free energy. He'll probably ask, what's the definition? And so when the ground state energy of the compound is G1, right? So that was what he drew on that graph. He had that little graph here, and he had it start here, right? And then it ended somewhere else. So say it ended here. So the ground state is that G1. If we went back to, what was it, chapter 3 or whatever, when we did here, right? So we could say like ground state versus end state or product. The ground state energy of the compound that is undergoing breakdown is higher than the ground state of the product. So here it's higher. This is our energy on this end here. So it's higher. And how we just calculate the difference is G1 minus G2 equals delta G. And that's all it is. All this is saying at constant temperature, constant pressure, constant pH, whatever. Don't really worry too much about that. He's not going to have you interchanging variables. We're not going to be at like 500 degrees Celsius or anything like that. Just know that this is at normal physiological conditions. Cool. All right. Laws of thermodynamics. Need to know them. He could potentially ask this. First law of thermodynamics is that the total energy in a system and its surroundings remain constant. So we can transfer energy, but we cannot. This is like we can neither create nor destroy matter, right? Same thing. We can neither. But for thermodynamics, is energy cannot be created or destroyed. It remains constant. It can be used. It can be released as heat, or it can be trapped making bonds or what have you. But we cannot make or destroy it from there. Our second law is that the total entropy of a system must increase if the process is to occur spontaneously. Right. So when he showed that diffusion example, right? He had, what was it? There was like the little square, and there was a line in the middle of it with all the gas molecules, and then when he removed that barrier, right, the gas molecules spread out. Right, so it's like a candle. You light a candle. After a certain amount of time, all the scent fills up your entire room. So entropy is just talking about like the disorder or randomness of the system. So the total entropy of a system must increase if that process is to occur spontaneously. Need to know those definitions. All right, and here's the equation that we showed. Need to know this equation for Gibbs free energy. It should just be in like letter form like this. Did y'all have equations on y'all's last exam? Which equations did y'all have? Y'all remember? The pKa? Henderson-Hasselbalch, was that on there? And was there one more or just that one? Michaelis-Menten was on there? Okay. So this is just one of those other ones that we got to know, similar to that. He shouldn't ask you to define any of them, just do you know the equation or not. But we've already learned that delta H is the change in enthalpy with the heat content. Delta S is the entropy, so the disorder randomness. T is absolute temperature, what have you. And just, he could, this is kind of like a, I guess it's very similar. So under some conditions, the enthalpy is equal to the change in internal energy. So just be on the lookout for, instead of an H, it being an E on the exam. Pretty much the exact same thing. Cool. Delta G is important because it gives us a lot of information. It's like when we're palpating, right? We're looking for our key markers, right? Is there any tightness, tenderness, hypertonicity of our muscle groups? Is the vertebra malpositioned or not? Is the correlating muscle from that nerve root, is it weak in any form or fashion? Is there wedging of the vertebra, right? Is one side higher than the other? So we kind of have those markers that we can get information from. It's the same thing. Like when we have a delta G value that's either negative or positive or 0, it tells us information about that. And those things are whether the reaction releases or requires energy, so whether it gives it off or receives it, the amount of energy, and the ratio of the concentrations of products to substrates. All right, so I'm just going to do a little bit of reading. Bear with me. I got this fancy thing. I found this, so it's going to be fun to use. All right, so there's a chemical reaction where we have our substrates producing a product, right? At equilibrium, we have roughly the same amount of substrates and products. Yes, at equilibrium, they're equal. So how we write that is arrows going both directions. Those arrows going both directions means equilibrium. Now, if the rate of the forward reaction is K1 and the rate of the backwards reaction is K2, then the equilibrium constant is determined as KEQ. So all that's saying is that KEQ is talking about the speed of the reaction of making products into the reverse reaction going back the other way. And at standard condition, it can be written as KEQ. I can't remember what the hell does that say. As KEQ. Is that a period? That's a period. Okay, that's not a multiplication sign. And it depends on the concentrations of products and reactants. So we can think of KEQ instead of it being K1, the rate of the forward reaction, in relation to the rate of the backwards reaction. We can just have it as products over substrates, right? So the amount of products that we have divided by the number of substrates. So if we have a lot of products, our KEQ is going to be very high, whereas if we have a lot of substrates, that reverse reaction is predominating, so that KEQ is going to be low. For thermodynamically, we can relate that KEQ with our delta G, with our Gibbs free energy. And this is equation number 2 that we have to learn. Did I have y'all learn this for the previous chapters? No? All right. So we need to know that delta G is equal to the negative RT natural log of KEQ. That's just, got to memorize it. And as always, as we saw up here, we can interconvert this ratio of products over our reactants in for our KEQ, right? It's just math. It's just, this is just a representation of that number. So, significance of delta G values. If the KEQ equals 1, then our delta G is going to equal 0. That means our reaction is in equilibrium. You might be wondering how they get these numbers. If we graph this on like our calculator or what have you, our natural log curve is, I was math club president, but I always forget like how the negatives, if it goes across the y-axis or x-axis. But a natural log curve kind of goes like this. And so whenever you plug in numbers, whenever you plug in 1 into this equation, the natural log of 1 is 0. So that's why delta G is going to equal 0. And then where if you type in a number greater than 1 into that equation along that graph, it's going to give you a delta G that's less than 0. And if you type in a number that is less than 1, it's going to give you a delta G of greater than 0. So if y'all want to have time and just play with that math, geekiness, really cool, I'll support you. Please feel free to do that. It'll give you a little bit better understanding. But if KEQ equals 1, delta G equals 0, we are at equilibrium. So we are making the same number of products as we are. We have the same number of end materials as we do starting materials, right, whatever we're starting with. If our KEQ is greater than 1, then our delta G is less than 0. So when our delta G is less than 0, that is our spontaneous reactions. That was the ball being rolled down the slide. We just had to put in that little bit of energy and it would go. The rate of the forward reaction is going to be greater than the rate of our backwards reaction. So we're going to have a lot more end products than we are what we're starting out with. So if we're doing glycolysis, we're going to have a lot more pyruvate at the end of that than we are going to have glucose as our starting material. We'll get to it. And then if we have KEQ less than 1, that means our delta G is going to be greater than 0. And all that's saying is that the reaction going backwards is greater than the rate of the reaction going forwards. So that would be we have a lot more glucose and we have like 0 pyruvate. Need to know that. He is probably going to ask you a question on any one of those, for sure. So he's going to say if the KEQ is equal to 1, then delta G is going to be positive, negative, or 0. And you'll say 0. Or vice versa. He could ask you if delta G is greater than 0, then KEQ is going to be 1. So it's just spending 10 minutes looking at that, understanding it. Not that bad. And going through these, when delta G is less than 0, it's negative. We are going to be releasing energy. So this was the breaking of our ATP. It released energy out. Energy is leaving. That's our exergonic type reaction. If that value is very high, or I guess if it's negative, very, very, very low, then the reaction goes virtually to completion and is irreversible. We have several things in our bodies. We have several enzymes in our body that catalyze reactions, right? We've talked about enzymes catalyzing reactions. There are certain steps that we want to do that we don't want to undo. In terms of glycolysis, One of those reactions is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1, PFK1. We've used one ATP up here, and we've used this second ATP up here. Pretty much we've invested energy into this process happening. We want to make sure we get return on our investment, right? So like when we invest in the stock market, we don't want to just say like, here's my money, like take it, thank you. It's like or when we go to Vegas, we want to like get money back. We don't want to just like have the house win every time. So there are certain reactions in our body that says, hey, okay, we're not going to go back from this. I've used this energy, I've invested, let's freaking do it, let's go. So that's all it's saying, is that when that delta G is greatly, greatly negative, you can't go back from it. One thing I want to point out is that our delta G value tells us nothing about the speed of a reaction. That's something that I know he asked on our test, and he should talk about in class, is that all it says is that that reaction is pretty much going to it's going to happen, and we're not going to be able to go back from it. It's not going to tell you how fast it happens. That reaction could happen in 5 seconds, 5 minutes, 5 hours, 5 years, 5 billion years. Like there's reactions that are still going on from millions of years ago on this Earth. It tells us nothing about the speed. In contrast, we have our delta G that's positive, so that's going to be our endergonic, or it's absorbing heat. The biggest thing is going to be associating endergonic or exergonic with positive or negative G, to be honest with you. And so this one, if the magnitude of the delta G is greatly positive, pretty much that reaction is not going to happen. For instance, I don't know what this paper is, but Form A. No, I'll turn it backside. What did y'all have Form A for? Did y'all have a test recently? All right. Diagnostic imaging test? Did EAA go well? Better or worse than the practical?
Worse.
I'm hearing mixed reviews. The class average was worse? Okay, so exergon for our endergonic, right? We're starting here. Our endergonic, we're putting energy in. So this is our energy on the y-axis increasing. If our end stage is all the way up here, right? We have to go just to get to there. It's so hard to come up with that amount of energy to get to that product that it never happens, right? So this is our 5 a.m. alarm that we set in the morning that says we're going to go exercise, right? It's going to take pretty much every ounce of energy that we're going to have to get out of bed, put those clothes on, and go work out, right? Or versus our exergonic reaction where it says, hey, this is the amount of energy I have to hit the snooze button, and so I'm just going to keep sleeping. All right, so if it's greatly positive, or greatly negative, greatly positive is it's probably not going to happen. Greatly negative is it's for sure going to happen. And 0 is equilibrium, no net change. No net change. All right, make sure, I've never really understood this, maybe one of y'all knows it, but whenever there's a little prime or circle at the end of the G, and sometimes there's like a hash mark too at the end of that, does anybody want to share? Okay, we'll go on. She'll come back, she'll yell at us. All right. Our endergonic reactions, right, we are putting energy into the system. Is delta G going to be positive or negative for endergonic? Positive, right? Delta G is going to be positive. we can actually couple those reactions to exergonic reactions. Exergonic reactions are going to release the energy, and the one for endergonic is to say, hey, thanks, like I'll take that and I'll go do whatever I need to do. So you got it?
G naught just indicates if it's at standard temperature pressure conditions. So
Okay, so G naught.
actually have that in a body environment because you're not going to be at room temperature inside the body. But it's standard temperature pressure. So 1 atm.
Do you know what the little tick?
The hash.
Yeah, do you have any idea about that?
Um,
What she said was the knot is just standard temperature and pressure. So like 1 atmosphere, whatever the perfect temperature is, what have you. It's just a lot of chemistry. I'm just saying, like, so if y'all see this on the test, I just use it as delta G normal. That's how I do it. But cool. So if whenever you see this, or whenever you, I think there's one later. Let's see if I can find it. If we've already passed it. There's one with a little tick mark on it too. I'm going too far. Anyway, we can couple exergonic reactions to endergonic reactions. And this is how we do it. The conversion of A to B. The conversion of A to B releases energy. Okay? So we're going from high energy to low energy. We've released some. So we can couple that to the conversion of C to D. So what's going to happen is this energy is released, and this endergonic reaction is going to pick it up and says, hey, I need this much energy to make that happen, and that energy is available. Boom, C is converted to D. As always, there is never a perfect reaction when it's releasing heat. We're always going to lose some as heat or as any other unusable form. Right? So our entropy is always increasing. If you think about it, when we work out, we're releasing some heat from our body. And so that's why we have this extra little bit of heat here as that unusable energy as a result of breaking down A into B. And that's just talking about the entropy is always increasing.
Can you go back and do that one more time? So A to B is releasing energy?
So our reaction from A to B is releasing that much energy. Okay? That is an exergonic reaction. So that is releasing that much energy into the system. This endergonic reaction from C to D requires this much energy. So what happens is it's going to use that energy that that B just released. It's going to pick it up and convert that into D. Right? It's going to reach that activation energy to make that reaction happen. And then this heat is just that leftover entropy from the reaction. Because energy is neither created nor destroyed, so we have to have this much energy at the start and that much energy at the end. So first law of thermodynamics. And then at the same time, entropy, the amount of randomness disorder, always increases, so that's where our heat comes into play. Whenever we break something down or we oxidize, right? Our oxidation, reduction, what number is that in our classification of enzymes? 1, right? Our redox reactions, what are those enzymes typically called? Dehydrogenases. Yes? Okay, so we have our dehydrogenase reactions. That's going to be termed catabolism. Whenever we're breaking something down or we have an exergonic reaction, that's catabolism. How I think about that, 2 different ways. The first one, whenever we have a major catastrophe, usually it leaves a lot of like destruction and waste to the area, so it like destroys a lot of stuff. It breaks stuff. And then also cats. I hate cats. I don't like them. They just jump around and like they'll knock lamps over or what have you. They'll come and jump on your table and like start eating your food. My roommate, like one of my best friends, had a cat, and that cat was awful. It was the worst, and it just like destroyed my life. So that's how I think it was a catastrophe that I had to be next to that thing. So those are exergonic reactions that are breaking stuff down. And our endergonic reactions are called anabolism. We're making stuff. We're making bonds typically with our anabolism. So catabolism, breaking bonds, releasing energy, like breaking the ATP into ADP and phosphate. Whereas anabolism is we're creating ATP, we're storing that potential energy within that bond. And how I remember that one is like that anabolism is like adding. You're adding things together, so you're making stuff. You're adding together to form a molecule. And those are endergonic. They pretty much all mean the same thing. Pretty much all mean the same thing. High energy phosphates play a major role in energy capture and transfer. What number was our transfer? What number would the transfer of a phosphate group be in our enzyme classifications?
2.
I heard one very loud, confident 2. I hear less. I hear some more, less loud, less confident, other things.
Hey, where's your candy? Come on, I finally get an answer right.
Definitely, I have been very bad about the candy recently. So that would be our transferase. So that would be our group transfer reactions. So we're transferring phosphate groups from one molecule to another molecule. And we can pretty much associate phosphates and our phosphate bonds with storing of energy. So when we break off a phosphate from ATP, releasing energy, we've talked about this. Actually, is it hydrolysis? Is ATP a hydrolysis reaction, or is it a transfer?
Hey, you said transfer, that's all
I think it's transfer. It's transfer. It's a transferase. But we can so what happens is we use water to break that bond, releasing the energy. What have you. 3 major sources of phosphate taking part in our energy conservation, which we'll be getting to here starting with chapter 9 with glycolysis, coming up pretty quickly. Glycolysis, citric acid cycle, and oxidative phosphorylation a lot of that is follow the phosphate. Where does it go? It's just a high energy molecule. And our last page, we'll get out early, unless you'll have questions. We can absolutely stay and answer those. Here's an example of coupling an endergonic reaction with an exergonic reaction. Once again, the first step of glycolysis. We're going to keep using it. Hopefully, y'all will know it very soon. You'll have 19 other reactions to memorize, roughly. Not that bad. Not that bad. All right. Reaction 1, glucose plus phosphate yields glucose 6-phosphate and H2O. This is the overall equation for our reaction. All right, so we're going to add a phosphate onto glucose, making glucose 6-phosphate plus water. Right? Do I need to go back and explain how that works, or can we know that glucose 6-phosphate just means there's glucose with the phosphate on it at the 6th carbon? Cool. That has a delta G of plus 3.3. Is that endergonic or exergonic? Endergonic, right? It's positive. Whenever we have a positive delta G, it's going to be endergonic. Is that anabolism or catabolism? Anabolism, we're adding, right? If it was negative, that would be exergonic, catabolism, catastrophe, cats. Reaction 2 is the hydrolysis of ATP with water, breaking that 3rd phosphate off, creating ADP and phosphate. That delta G is negative. So delta G is negative, exergonic, catabolism, right? catabolism. If delta G is less than 0, what is the KEQ? Greater than 1, right? Being able to interconvert all those things is highly, highly important. So if it's greater than 1, delta G, you can just say it's opposite, right? If greater than 1, delta G is going to be less than 0. If KEQ is less than 1, delta G is going to be greater than 0. But just making sure you know where that 1 and that 0 is at. This reaction has released 7.3 calories per mole, whatever. This one's got 7.3. This one needed 3.3. What happens is we're going to break that ATP bond first, releasing that energy. This reaction, I don't know why he has this as number 1, but it's okay. This should probably be number 2. This reaction needs 3, so it's going to take that energy from that breaking of the ATP bond, and our sum of both of those reactions is a delta G of -4. Overall, both of those are an exergonic reaction, overall net, because we released 7 here and we only needed 3 on the other side. So that's why this reaction occurs. It's spontaneous because the delta G is less than 0. Okay, you won't have to do any math on the test. This is just kind of learning, obviously. Everything's learning that we do here at school. But just for your own benefit. And that enzyme is hexokinase. All right, let's see. This was a test question that I did not expect. All the way down here. There are other nucleoside triphosphates, right? So instead of adenine triphosphate, we can have other nucleosides here, and they can transfer phosphate groups as well. Right? We've seen GDP, right, and GTP with our G protein coupled receptors. We also have U for uracil and C for cytosine. All right, cytosine. All right, we learned that from our amino acid, from our, what was that? When we do DNA, RNA, right? It's the same ones. So I think the question that he asked was, which of the following cannot participate in group phosphate transfers? And he just had, the question was like UDP, GDP, CDP, and like RTP. You pretty much just had to know that RTP was the wrong one. So look for the ones that are in our DNA, RNA, and you'll be good. It's just for the people who didn't study. They're looking at all these letters like, what the hell is this? Like, I didn't read that last little paragraph at the end of the chapter. And that's pretty much it. We're 2 chapters ahead of him now. You don't believe that? Are there any questions? Comments, concerns, compliments?